Pharmacokinetics PK assays are critical tools used to evaluate the behavior of drugs in the body over time, providing essential data on absorption, distribution, metabolism, and excretion, ADME. These assays support drug development by helping to establish dosing regimens, predict efficacy, and identify potential toxicities. Key components of pharmacokinetics assays. Absorption determines how a drug enters the bloodstream, often studied using CACO2 cell models or in situ perfusion. Distribution examines how a drug is distributed across tissues and organs, commonly analyzed using plasma protein binding studies or tissue partitioning studies. Metabolism identifies how a drug is chemically transformed, often by liver enzymes. In vitro assays like liver microsomes or hepatocyte studies are commonly used to assess metabolic stability. Excretion studies the elimination pathways, including renal and biliary routes. Urine and feces analysis are typically employed in excretion studies. Common pharmacokinetics assay types. Plasma concentration time studies measure drug levels in plasma over time to calculate parameters like half-life, Cmax, maximum concentration, and AUC, area under the curve. Enzyme kinetics assays assess the rate of drug metabolism using enzymes like CYP450. Transporter assays study the role of transport proteins, for example, P-glycoprotein, in drug uptake and efflux. Bioavailability and bioequivalence studies evaluate the fraction of an administered dose that reaches systemic circulation. High-throughput screening, HTS, rapidly screens large numbers of compounds for pharmacokinetic properties. Drug-drug interaction assays examine the impact of other compounds on the drug's pharmacokinetics. Techniques used: LC-MS/MS, liquid chromatography, tandem mass spectrometry. Gold standard for quantifying drugs and metabolites in biological samples. HPLC, high-performance liquid chromatography, separates and measures components in a mixture. Radiolabeling techniques track drug distribution and excretion using isotopes. Fluorescence or UV detection for compounds with suitable properties. Applications optimize drug dosing, identify potential risks of toxicity, ensure drugs meet regulatory requirements, FDA, EMA, etc. Support translational research from preclinical to clinical trials.